Hello and welcome to this evening's Zoological Society of London event. My name is Charlotte and I will be your host tonight as we discuss how we can recover extinct in the wild species. We've got four fantastic speakers lined up for you tonight and they're all eager to take your questions. So with that in mind, I'm just going to share my screen to show you how you can do that. During tonight's event, please do submit your questions for our speakers. You can do this in two ways, either by going to Pigeonhole, so that's www.pigeonhole.at/1931. That's www.pigeonhole.at/1931. And there you'll be able to write your question and submit it to us. Most importantly, you can also upvote other people's questions, and I will be keeping a close eye on this this evening because I'm expecting there to be hopefully lots of questions, and I'll be prioritizing them by the most popular questions. So please do upvote anything that you would particularly like to see answered. The other way you can submit questions this evening is by email. Our address is scientific.events@zsl.org. That's scientific.events@zsl.org. And as always, at the end of the event, I will just mention it now, we really welcome your feedback as well. We'd love to know what you thought of tonight's event. So please do go to the SurveyMonkey survey at the end of the event to let us know what you thought, but I'll share that link with you again later. So as I mentioned, we've got four fantastic speakers lined up for you tonight. And our first speaker is coming all the way from Canada. His name is Dr. Axel Merhenschlager. Axel leads community conservation and conservation translocation programs as Director of Conservation and Science at the Calgary Zoological Society. He is also chair of the IUCN Species Survival Commission Conservation Translocation Specialist Group. So he's going to be introducing the topic this evening and discussing the ultimate test for conservation. Axel, thank you so much for joining us. Over to you.
My pleasure, and thank you so much. It's really a privilege to be joining all of you today. And I'm going to be talking to you about something that I see as one of the greatest challenges of our time, which is not just how to save species, but to save those species that are most at need of our help, species that are extinct in the wild. So first of all, I'd like to very much acknowledge my co-authors in this, who are joining me to present the ultimate test for conservation. Indeed, this comes from a whole number of organizations in a task force that we have formed through the IUCN Conservation Translocation Specialist Group. And these are actually expanding specifically to take into account, the vast biodiversity of plants, which we very much embrace and want to ensure that along with animals and fungi, that we advance their conservation of all species, including those that are extinct in the wild. Now, there are species on this planet that would not be here if it wasn't for effective conservation translocations. For instance, the California condor is a species that went down to just 27 individuals until the last ones were caught up in 1987. And due to the amazing collaboration of many organizations, are now flying again in the skies in their hundreds. Here we have Rabs's fringe-limbed tree frog, which is amazing because it can actually spread its appendages to float through the forests of Panama. And this specific individual is called Tuffy. And Tuffy is the last male of his kind, having been rescued in light of the deadly chytrid fungus that has affected so many amphibians. As he leaps off, and as we look at this footage, we now remember that years ago he passed. And with that passed the last known individual of this species, which is likely extinct. So here we see the cornerstones of possibilities between success and between failure. And when we speak of failure, we need to address the immense challenges that lie ahead of us. Extinct in the wild species are so because of things having gone terribly wrong in the past. For example, for the black-footed ferret, the only native North American wild ferret, it actually was thought to be extinct in the world until the last ones were discovered on one ranch in Wyoming in the 1980s. Thousands have been bred by zoos, but actually the ecosystem receiving them is in trouble. The black-tailed prairie dog, which is its primary prey source, has gone from 98% of its range. In Canada, through years of planning and deliberation and collaboration with the Americans, with Calgary Zoo, with Parks Canada, we were able to have the first releases of black-footed ferrets go back into the wild in over 70 years. And indeed, they did very well. The survival rates were very good. They reproduced. We even had grandchildren produced within 3 years. So you'd think the world is terrific. However, within this ecosystem is an invasive species. In fact, a species which is known for the Black Plague or sylvatic plague in animals. And here in green from a recent paper, you see sites where we actually have plague-positive fleas in prairie dogs or in Richardson ground squirrels. And the red crosses are actually of prairie dogs that have died of plague. So we've been working on how to mitigate this, but it's a big challenge. The other challenge is that prairie dog populations have actually been dropping in response to drought, in response likely to climate change. So here we are trying to recover a species in light of immense disease and in light of climate change. And that actually led to a temporary stop of reintroductions of black-footed ferrets until we can address these challenges. So you see, these efforts are not easy, but it is possible to have successes, like for the Przewalski's horse, which actually went down to just 12 individuals left in zoos in the 1950s and one caught from the wild. But now, due to, again, many amazing collaborations, they roam free in their hundreds across Mongolia and across China, with potential reintroductions in Russia and Kazakhstan coming. So the possibilities are tremendous, and they are needed, because even in the IUCN Red List, we see that there are species that are at great risk of extinction. For instance, amphibians, at 40% of described species at risk of extinction. And even if we account for those that are not yet described, then we see that there might be up to 1 million species that are close to extinction, and it isn't just capturing the attention of environmentalists anymore, but even of economists, for instance. Conservation translocations are very important and have been growing 30-fold across 30 years in response to trying to save endangered species and to help ecosystems that need that kind of support. Recently, a consortium of organizations put forward a motion for the IUCN World Conservation Congress, which will now happen this fall, to support global support or to efforts to bring back extinct in the wild species. So of 115 government organizations, 95% back this, and 99% of 572 NGOs around the world have supported these efforts, which we'll talk about more right now. There were 5 key components in the motion. And the first one was, first of all, encouraging and applauding IUCN for its amazing red listing initiatives and furthering those kinds of initiatives over time, but also asking to describe and to consider populations that are potentially formed outside of the indigenous range of species and consider how those can be useful for the recovery of those species. We'll talk about it a little bit more. When we speak of extinct in the wild species, currently there are 42 described plant species, and they are primarily in terrestrial systems. And among animals, there are 38 species extinct in the wild under human care, and these are from terrestrial systems, but also from freshwater scenarios. Interestingly, there are also species that are possibly extinct in the wild that we've been looking into further. And these are species that may or may not have healthy populations under human care and may or may not still have individuals left in the wild. The interesting situation is that among conservation translocations, there is a scenario of releasing species beyond where they've ever been, normally to avoid extinction in their indigenous range. For instance, if they just can't escape the threats. And I helped to workshop some scenarios at an international conference that we hosted in CTSG in Chicago in 2018 on Lister's gecko and blue-tailed skinks. These are home to Christmas Island off of Australia. And just in the last 12 months, blue-tailed skink have actually been released onto a different island group where they've never been before, where they're doing very well, which may be key to helping them be safe from extinction, but in a way where one needs to be very careful as to the impacts on receiving ecosystems. Review that we just have coming out on terrestrial arthropods also shows that actually 16% of conservation translocations now are involving assisted colonization. And we are encouraging that these techniques be looked at very thoughtfully and carefully, and in a way to see whether and how and when they can be useful for the recovery of species and for ecosystem processes. We're asking for not just plans and not just talk, but for action. We are asking for plans to be established for all extinct and wild species by 2030, and we want to see and help achieve significant demonstrable progress by 2024 as a significant contribution towards achieving the CBD post-2020 strategy for biodiversity. Indeed, when we look at species, specifically animals, for instance, we ask the question, are all those species that are under human care actually a sufficient number and quality and distribution, if you will, to be an assurance or insurance population for the future? And interestingly, we might take, for instance, a benchmark of wanting about 500 individuals, such as the top line that you see here, to have an effective population size that is conducive for return to the wild. But you see that across mammals and snails and reptiles and birds and amphibians, you actually have populations that are at quite low number. And so we need to look critically at the status of these and how we can improve them. In fact, considering ex situ populations is very important, and we advocate for looking at it more closely in the red list context as well. This, for instance, is a mountain bongo. It's an endangered subspecies that my research group works on in Kenya in collaboration with Kenyan agencies. There are fewer than 60 left in the wild, but there are actually hundreds in ex situ populations around the world. And so taking this into consideration is important. What's also important is to look at the institutions that are involved. Back in this case, for extinct in the wild species, for instance, you might be shocked to find that several of these extinct in the wild species are only in one institution, or two, or three, or four. And so that actually puts a big onus on those institutions. Now, remember that within the pandemic that we've had recently, is that there's been tremendous pressure on botanical gardens and on zoos because of, them being closed, because of pressures on their finances. And so ultimately, the viability of those institutions is linked to the viability of those species. And this is something that we look to draw attention to last year in a paper in Science with a bunch of authors, including John Ewan here. We also urge that these activities for extinct and wild species be in alignment with the IUCN guidelines for reintroductions and other conservation translocations. These, of course, are well established. We launched them in 2013. We've been translating them into many languages so that they can be used around the world. And indeed, they are implemented into policies, such as with the Scottish Code for Conservation Translocations, such as under the Council of Europe, in terms of adopting the guidelines for conservation translocations under climate change, or in other country contexts like in Canada, where the Canadian Wildlife Directors Committee calls for these kinds of, or for the guidelines to be implemented by any of the government jurisdictions, by any of the agencies. And we've even gone further than that, because as we look at it, extinct and wild species, we might think about those that have already gone extinct, and indeed of efforts that are currently happening to try and bring some back, driven by some organizations to do so. And so we first of all published a paper along the lines of adapting the IUCN guidelines to ask critical and important questions of what is needed for conservation. But then we were also asked to come up with guidelines for the world on de-extinction, which are called the guiding principles on creating proxies of extinct species for conservation benefit. So we are well positioned in terms of our knowledge. And we have so many species to draw from too. Aside from the primary literature, we have now the 7th book that has just come out last week, spearheaded by Pritpal Saray, on the global conservation translocation perspectives, outlining hundreds of species across all biomes and ecosystems that you can imagine. So fourthly, we urge zoological and botanical gardens to lead the public awareness of the plight of these species, and also to help in providing actual individuals for release when the time is right. In my research group, we did a systematic review of animal conservation translocations in North America in 2016. And we followed that up with that, and that was for 279 species. And we followed that up with asking, actually, how relevant are zoos to the conservation translocations that we know go on? And about half of the translocations involved releases from human care. But among those, actually, zoos were only involved in 1/4 of those conservation translocations. And so working for a North American zoo, I think I'm safe to say that we were pushing for zoos to work collaboratively and for others to support them more so that more individuals can get in the wild in responsible ways. Indeed, time is ticking on the suitability of many of these species. And not to steal Amanda's thunder, who will speak shortly, we have a paper that's just come out that's showing multiple life stage inbreeding depression on a species called the seaheck or Guam kingfisher, which has been extinct in the wild now for over 30 years. And so it's not that you can hang on to these treasures forever to just one day put them out. The time is ticking for action. We call on donors to support efforts that aim to assess the status of species according to the IUCN Red List, and especially to help resource efforts that work to reestablish species in the wild. When you have the right alignment and the right support, amazing things can happen, such as for the scimitar horned oryx, which was extinct from Canada for decades. And through the backing, especially of the United Arab Emirate government, has now made a return back to Chad, and in a way that's quite impressive, both in scale and in scope so far. There are questions to be answered, and our task force will engage in answering them over time. What species are currently extinct in the wild, and which ones are coming, which will likely be so soon? What are the causes for species to become extinct in the wild across taxa? And what is the status of these ex situ populations, not just now, but in the future? What barriers prevent extinct in the wild species from returning to the wild? Where and how could such species be released responsibly? And what lessons can be learned from the recovery of past extinct in the wild species that we can apply now and into the future? Several years ago, I had the privilege of seeing Sudan, the last northern white rhino in Kenya. And the garden, I spent over an hour with him. And even though he wouldn't have known any different, for me, it was very significant. This is a species that we failed. And being a zoo animal and quite tame, I put my hand on him and I apologized on behalf of all of us because we had let his species down. And in this way, I was looking extinction in the eye. And I don't want to ever do that again. And so really the onus is on us to change it. And to change it not just on behalf of the species, but on behalf of people that care. For instance, this man on the right-hand side who's in a workshop in Calgary that CTSG helped run for the seaheck, the Guam kingfisher. For his indigenous Chamorro people of Guam, this is a culturally important species. And he is a conservation hero, having been one of the 2 people that caught the last individuals from the wild and who hopefully will be among the first to return them to the wild in the next couple of years. It is not just about species, but people, like for this First Nations dancer in Canada that danced in genuine joy at the return of the black-footed ferrets back to the prairies not just because of the ferrets and not just because of the species and its viability, but because of the connection between that species and the ecosystem, and his hope that with that return, so would return the landscape ethic of his people and a resurgence of their culture. Here we see Spixus macaw, a beautiful species that actually became the subject of a Disney animation feature called Rio. So Spixus macaw, is that a species that we're going to remember as a cartoon character? Or are we going to remember it for returning to its native rainforests in Brazil? Extinct in the wild species are special to me in many ways. They are the ultimate test for conservation. Unlike those species that have not yet been described, or those species that pass away silently in forests or in coral reefs somewhere without any much attention, these species are in our care. They are the ultimate conservation refugees. They will live or die as a species on our watch. So with them, in a way, I think they're the greatest opportunity for us to show that conservation is powerful, that conservation works, that a collaboration between agencies which runs from school children to educators, to scientists, to advocates, to politicians, to funders, can achieve great things when working together. So I ask you to join us now and in the long and difficult road in the future to make a difference. Let's do this for their sake and for ours. Thank you.
Axel, thank you so much. That was a really powerful introduction to the subject, and the questions have already been streaming in. I am just going to remind everyone one more time of how you can share your questions with us this evening. You can either go to www.pigeonhole.at/1931 to submit your question. And if you do that, then please, if it's for all of our speakers, that's fine. But if it's for a specific speaker, do sort of mention their name, and then I'll be able to spot who you'd really like to answer that question for you. The other way you can submit questions is via email, and our address is scientific.events@zsl.org. So Axel, thank you again. We will now try and get through a few of these questions before we move on to our next speaker. The most popular question so far, Axel, and I think one of our other speakers, Paul, will hopefully have something to mention about this later. But the most popular question so far is, how can we get society to focus on bringing animals such as insects and amphibians back into the wild, which are just as important, if not more, than the mammal species, which society focuses on more. I'm sure this is something you're very aware of. It's like we do tend to focus on sort of the big and the charismatic and forget about some of the smaller stuff. What do you think can be done to address that?
I think there are a number of things that can be done. And I really love this question, by the way, because it's something that was on our minds. And that's why we put together this systematic review recently. And we were actually pleased to see that there, are about 130 species that are involved in terrestrial and conservation translocations alone for arthropods. And the most popular ones actually being butterflies, but also other ones which are very evolutionarily distinct, like the weta, for instance. I think one of the things that we need to come to understand worldwide, and I think people do, and often children especially do, is we need to understand the interconnectedness between ourselves, the air that we breathe, the water that we drink, the food that we eat. Where does it come from? It comes from nature. So when we think about insects, for instance, what is pollinating those crops? It's insects that work for us, giving us ecosystem services for free. And we would let that go extinct. We would let that pass away. It's not fair in some ways, morally, ethically, to those species themselves, but it's also not strategic, even if we only care about ourselves.
No, very, very good point. In your talk, Axel, you mentioned the northern white rhino, and there's a question relating to that, which I think is very much trying to sort of make the point as to how you prioritize what you focus on, and what you spend your money on, because you know, there has been quite a lot of coverage about the northern white rhino. There is a fair amount of money going towards trying to conserve it, and yet there are only a couple of individuals left. Do you find yourself having to sort of justify what you focus on, which species you do decide to try and bring back into the wild?
It's very interesting. In our research group, actually, we're working very hard on a current research paper that evaluates the benefit that we can bring for species, the cost, and the likelihood that others will help to support that cause. One of the things that I think is a misconception is the view that somehow the world is united in overseeing a finite amount of money to put towards conservation that we as a society or as a global community can make decisions about. That's not the case. There are different sources of money. And so if there are supporters that will support certain initiatives, but not necessarily others, why wouldn't we embrace that? I think therein lies the key. And for me, the key really lies in expanding that. I deal with government, I deal with philanthropists, you know, nationally and globally. And basically, it's about engagement and inspiring those groups to really make a difference. I think that we can save, or we can try to save, you know, charismatic megafauna at the same time as charismatic mini fauna, as the invertebrate specialist group calls it.
Fantastic. I'm going to try and take one more question before we move on, Axel. Another popular question that's come through is about how do you collect data from data deficient countries regarding their wildlife? So countries, for example, that might be war torn, it's very hard to collect that information, and so there's a question around that, and also how these countries and low-income countries can be supported and helped.
I did my PhD through the Wildlife Conservation Research Unit at Oxford University. And I remember learning at that time, coming from a rich continent, if you will, that learning from others, that in many places in the world, wildlife that does not pay for itself will not exist. And so I think what is really crucial is to make sure that one engages those countries and local communities in a way that is economically advantageous for them in the short term and in the long term. Meaning that it isn't just Western scientists that go there to collect data, but let's involve, for instance, I work in Ghana on a hippo sanctuary, which won a United Nations Equator Prize for community conservation, where we actually have engaged and trained local people that can identify over 250 species of bird by song. And it is in a way that has yielded now schools, fresh water, lighting, and everything like that in concert with assessments of biodiversity, helping hippos, helping birds.
Fantastic. Well, thank you very much, Axel. We will be bringing you back for further questions at the end of the event. So do keep sending your questions in for Axel and for all of our speakers this evening. And as I mentioned, do include their name if you'd like your question to go to a particular person. Our next speaker, though, this evening is going to be talking about plants, which is very, very exciting. We often focus on animals here. So we're joined by Dr. Sarah Dalrymple. Sarah is a senior lecturer in conservation ecology at Liverpool John Moores University. She is a plant ecologist focusing on threatened species, seed ecology and conservation, and the efficacy of conservation translocations under a changing climate. So Sarah is going to be discussing how we restore extinct in the wild species. Sarah, thank you for joining us.
It's a real pleasure to be here. I'm just going to share screen. It's quite a responsibility to be representing the plant world. But I'm really pleased to be here. So thank you for asking me to speak about these very interesting and important species. So my talk today is going to focus on some of the 42 species of extinct in the wild plants that Axel mentioned, but it's also going to rely on some studies that me and collaborators across Europe have been working on, which have come out as a paper. So we had a conservation biology paper last year that came out on ex situ collections. And by ex situ, I mean for plants, that means botanic gardens, so living collections in botanic gardens, and also seeds kept in seed and gene banks. And we also pulled together a load of brilliant studies for the Journal of Nature Conservation and wrote an editorial for that. So if you're interested in following up any of the studies, nearly all of them have come from one of those two sources. So what I'm going to talk about are some of the 4 golden rules of good translocation practice. And that includes selecting good founder materials. So that means for us plants and seeds that have been are going to be outplanted into the wild. Identifying optimal habitat, eradicating or minimizing the threats in that habitat before we put the release propagules or seeds or plants out into the wild. And then our final aim, which is the main aim, to create a self-sustaining population. So I'm going to start with my first one, which is selecting suitable founder material. This example is Rubus humilifolius, which is a member of the bramble family. You might recognize the leaf shape and the flowers there. And this was collected from its last known location in Finland, just hours before the population was decimated by development. So when they wanted to reintroduce it to the wild, they'd moved it to a garden where it'd been growing, but in very, very small numbers, and they had to micro-propagate it. So that means like quite a high technology intervention to try and get enough plants to release into the wild. The good news is that it has been reintroduced into the wild and is doing quite well, but that investment that's needed for micropropagation might not be available for many species. And it's also, it's a lot more than just collecting some seeds, growing them for a bit and then putting them back out into the wild. It's quite a technical fix. So if we look at a data set of these 42 extinct in the wild species, you don't need to worry too much about what the species names are, but you can see them all represented in different colors. So if you look at the scale to the left-hand side of the screen, you can see that it goes from 0 up to, well, it's 110. You can't actually see it on the scale. But that represents the number of ex situ collections that these species are held in around the world, according to the Plant Search database. Now, some of them in dark red are actually represented in 0 collections, which can't be true, and tells you something about the need to collaborate and share records. But the sort of main message that you can see from there is, just as Axel was saying, a lot of these critically threatened species are actually held in very, very few collections, which means that we might need to intervene using the sort of more high-tech interventions to actually bulk up their numbers for release. And the second example, this Eriocolon species, which is a wetland species in Japan, this was reintroduced into the wild after several decades of being in a botanical garden. But the reintroduction failed because the species had acclimated or got used to the botanic garden conditions. And again, if we look at the species, we can sort of see that this might be the case for lots of our extinct in the wild species, especially when you look at the amount of time that's lapsed since we last saw them in the wild. So this timeline again shows our 42 species. It's been color coded according to the longest out of the wild, which is the Franklin tree, and that was lost back in 1803, which hopefully you can see my mouse just there. And then we follow that a jump to 1905, but all these species were lost in the wild before that Eriocolon species was last lost from the wild. And even for the ones that have more recently lost from the wild since like the 1990s, the chances are that they could have acclimated to botanic garden conditions as well. So there's a chance for maladaptation, which means it's not suited to its in situ or wild conditions anymore. The second sort of golden rule of translocations is to identify optimal habitat. So in this example, actually, this is a really nice example of how plants give us a little bit more leeway than animals. So this was another species lost in the 1800s, Erica turgida, and the map shows an area around Cape Town, which is now highly urbanized, but was this species quite narrow endemic range. They managed to cultivate many hundreds of Erica turgida and used that number of plants to put out into habitats which actually they weren't convinced was going to be suitable. But the good thing about introducing the species into what we might think of as marginal habitat is that they could actually learn a lot from what the species was adapted to. It's such a long time since we saw it in the wild. We can't make any observations as to like the micro habitat where the seeds might germinate and all those sorts of considerations. So it's quite useful to be able to try these trials reintroduction where we think that they might not work, but learn from that. And like I say, you can't really do that with animals. There are welfare issues to consider. And what happens if we can't raise hundreds of plants for trial translocations? Well, we can use predictive methods. And my PhD student who's just handed in his thesis, Joe Bellis, he wrote this paper which was in Conservation Biology again last year. And this sort of workflow shows how you can take the distribution data from threatened species. In this case, it's a butterfly that we're working on. And there's many hundreds of distribution points there. You can add it to the climate data and then produce projection maps which tell us where the suitable climate is likely to be. Now, this is current climate, so then we could say whether translocations were going to work or not. But for this sort of technique to work, you have to have at least 30 sites, and the more the better, really, to have confidence in the predictions. So my next figure shows, again, our 42 extinct in the wild plants. And what it's showing is the number of geospatial records which we can download from the global databases. In this case, it's GBIF database. So that dark red bar at the left-hand side of the scale shows that about 30 species are only known from 50 or less geospatially recorded occurrences. And actually, a lot of them are from 10 or less different records. So we can't use those predictive models, and that means that maybe trials are going to have to be used at great expense. Now, if we look at this map of where all these species are from, you can see that the restricted range is explained by the fact that quite a lot of them are island endemics. So they're, even if they were at their fullest extent that we could look back in time to, they would still be in just a few square kilometers of habitat. So that just means that, again, some of these predictive methods just won't do much good. And we might not be able to track habitat suitability over time. Okay, so on to my third point, which is that we want to eradicate or minimize threats. And if I just give you this example, which is Cyanea superba, which is a Hawaiian plant species that was extinct in the wild. They've done a really amazing job of bringing it into cultivation, raising it from seed, and then putting many hundreds of plants out. And according to this report, again, this is one from our Journal of Nature Conservation special issue, they'd managed to get 900 or so of the plants to survive in the wild. Now, you probably know about the threats on Hawaii. It's mostly from invasive species, things like rats and rodents, other rodents that are eating the seeds in this case. There's also problems from pathogens, all sorts of different things are going on for these plants. And what it means that although they've got good survival, they haven't necessarily seen good seedling survival from seeds germinating in situ. So that's the seeds dispersed from those plants. Now, what they have seen is that survival is better if you move the plants upslope. So technically, it's assisted colonization, which, again, Axel alluded to earlier. So that's moving things out of their historic range. Now, on Hawaii, when it's a volcano, it's still technically within the range boundaries. It's just into an altitudinal zone that maybe we didn't know the plant from before. So maybe in lots of cases, it's not going to make people too upset. But it still is something that needs considering. And when I look at the threats for our 42 species, which includes Cyanea, in fact, but if this word cloud is showing what is generated from the threat evaluations that are in the red list, and you can see the usual suspects are appearing from the threat evaluations. So we've got invasive species. Feral pigs get their own special mention because they're particularly problematic, but also things like agricultural intensification, deforestation, lots of things that would be familiar to anybody that was at all conversant with the current sort of environmental plight that we have at the moment. What's not mentioned in this word cloud, and when I went through it with a fine-tooth comb looking at all these different evaluations, is climate change. So for Cyanea, they suspect that actually moving things upslope is going to be finding some optimal habitat. But in most of these existing red list profiles, climate change isn't mentioned at all. So it's something that we really need to do something about. So on to my final section, which is aiming to create a self-sustaining population. I've just got 2 examples to show you. So these are 2 grass species, 2 bromes, and these are associated with the high disturbance that you find in traditional agricultural practices. Now that through seed cleaning and pesticide application and all these different, and also intensifying the crop cycles, these species have been lost. So they were actually, I think one of them was found in a packet of seeds somewhere. And one of them was discovered in a garden. So they were both classed as extinct at one point. And both the authors of both the papers that are cited here are not confident about the outcomes for these species because they rely on continual management. So, what does that tell us about the prospects for these very highly threatened species? So this is my final slide. I just wanted to say that I don't want to be too doom and gloom, but at the same time, these interventions, we can translocate and restore species that are extinct in the wild, but it's very difficult. And my collaborators and colleagues on the Conservation Translocation Specialist Group, they're represent loads of experience and knowledge, but we're not magicians. So I just wanted to say that it's really important that we act in situ as well as considering translocations, because that's really how we're going to stop extinctions from happening. And that's the end of my talk. Thank you.
Thank you ever so much, Sarah. Oh, I'm just going to try and turn my video back on. There we go. Thank you, Sarah. That was fantastic, and we have had some questions through for you already. I will also mention there's one question I can answer, which is whether this event will be recorded and archived afterwards, and it will. It will be on our YouTube channel, so you'll be able to find it there again in the future. But Sarah, the questions for you, as I said, we've got several that have come through, and one is whether you believe that the rising global average temperature will provide challenges for species whose seeds need cold stratification before they're able to germinate.
That's a fantastic question. And actually, although a lot of the species I've just been talking about are tropical species, I'm a cold environment person. So I've done quite a lot of work looking at the requirements for cold stratification for seeds. And we do think it's quite an important mechanism for community maintenance in alpine regions and montane regions. They do require chilling. And one of the things that happens is it's quite complicated, but in certain areas, actually the loss of snow beds actually makes the temperature cooler. I know it can sound a bit counterintuitive, but we're quite worried that the insulating effect of loss of snow beds particularly at high altitudes and high latitudes means that the seeds actually aren't very well adapted to the conditions that they're used to, and that allows a community change. So some of these species are very threatened, so they might class as critically threatened, but what we're particularly concerned about is the community level, because we'll see a change from the sort of alpine floras into things that are more associated with boreal forest and things like that. So that progression will sort of change upslope. So yes, it's a little bit off topic, and actually I could give you a whole talk on that. But yeah, it's a really good question. It's definitely something that the seed conservation community are very worried about.
Another question that came in quite early on, Sarah, was sort of the role that maybe people can play in their own gardens. So the question is, would it make sense to try and see if people are able to grow plants in their gardens and collect seeds or saplings to help boost the number of endangered species? Is this something that can be of help and of use?
Yes, definitely. I think so, but it has to be done under very, I suppose, carefully planned situations. It's actually, I haven't planted these questions, by the way. These are excellent questions. One of the papers that I said about the special issue from Journal for Nature Conservation actually wrote about this, because there's a national park, I think it's Majella National Park in Italy. I'm probably not saying it right, actually. where they have actually they've given out plants to local homes and local schools to increase the amount of plants that are in domestic settings. And that's increased that sort of pool of genes and basically increased the resources that are being devoted to growing these plants. So yeah, it is something that people are looking for, although it's very novel. That's why it was in our special issue last year, because it was the only example that we knew of. So yes, but it's a great idea, I think.
Oh, okay. Might perhaps see more of that in the future. And one last one for you, Sarah. A question which is, if a species, oh, sorry, I've lost it here. Here we go. I'm sorry. What do plant conservationists consider, or do plant conservationists consider that the last place a plant species is found may be a refuge rather than its prime habitat? I mean, could it be that this plant has basically been pushed to its limits?
Amazing questions. So yes, absolutely. There used to be that sort of idea that, oh, well, if we'd lost it from the wild from this location, we should try and put it back there first. And actually, that's exactly right. These habitats where the plants and animals, in fact, this is true for, have been sort of squashed to, that's just the last bastion that they've held on to. It doesn't mean it's optimal. So yeah, it's quite important to look at very long-term range records, not just the last few decades, and understand what the tolerances of the species might be over much longer time periods for that very reason, because there's lots of examples where that's happened.
Brilliant. Well, thank you very much, Sarah. We will bring you back later on. But for now, we're going to move on to our third speaker of the evening, who is from the Zoological Society of London. We're joined by Dr. Amanda Trask, who's a conservation biologist within the Institute of Zoology at ZSL. And she has a particular research interest in population ecology and genetics. Amanda is part of an international team working on recovery planning of the extinct in the wild sea heck, also known as the Guam kingfisher. And so she's going to be telling us more about this now. Thank you, Amanda.
Hi, yeah, it's great to be here. So I'll just share my screen now. I'm going to talk today about recovery planning for an extinct in the wild species, the sea heck or Guam kingfisher. So sea heck are endemic to the island of Guam. This is in the Western Pacific and is a US overseas territory. But sadly, they were extinct in the wild after the accidental introduction of brown tree snakes to Guam in the 1940s. So brown tree snakes are predatory, and they predated a lot of the native bird species of Guam, including the sea heck, so that by 1988, the sea heck were extinct in the wild. However, luckily, before their extinction in the wild, 29 individuals were able to be caught and were translocated to zoos in the US mainland. And after that, a breeding program was able to be set up, so that currently there's a population of about 140 birds in zoos in the US mainland, as well as back on Guam in the Department of Agriculture. However, although 29 birds were able to be caught from the wild, only 16 of these actually successfully bred. So the current population is founded from only 16 individuals. And we know that small populations can be at risk from inbreeding. So this is where related individuals breed together to produce inbred offspring. And these inbred offspring can have reduced probability of survival or reproductive success, and that's termed inbreeding depression. So we were pretty concerned about inbreeding depression occurring in the sea heck population. So we looked at this using studbook data provided by the different breeding facilities that were breeding the sea heck. And we did find evidence of inbreeding depression in the sea heck. So we found that individuals with high inbreeding levels had shorter lifespans on average and also had lower reproductive success than more outbred individuals. So this was effects that we'd found at the individual level, but what did that mean for the population's viability? So that's what we wanted to look at next. So we created population models. So here you can see population size on the y-axis, and the population is being projected forward through time. So the blue line shows a scenario of the population under current management and with inbreeding depression included in that model. And you can see the population is projected to decline. So that's pretty concerning. And then we also simulated a scenario of removing individuals from that population for releases to the wild. So that's that black dotted line that you can see. And we found that actually removing individuals for release to the wild could actually increase the rate of decline. So the population might not be able to recover from those removals of individuals. So that's really concerning because we need to remove individuals to recover the species to the wild. So we talked to conservation managers to see what we can maybe do about that. And we came up with a scenario of increasing breeding in the population. So if you see the blue line on this graph now, that's modelling the population with that increased breeding. And under that scenario, we could actually increase population size to 200 individuals. And if you look at the dotted line, so that's the same scenario as before, removing the same number of individuals for releases to the wild. And we find that actually the population could support those releases now. So that's good news, because if we can increase breeding in the population, then we can recover the species back to the wild. However, if I draw your attention back to the blue line, this is 200 individuals, so that's still not a very big population, and that reflects the limited space available in zoos. So the sea heck is expensive to keep in zoos. It has a specialised diet. They're also quite territorial species, so they each need to be in an enclosure, each of themselves, or potentially as breeding pairs. So the zoos can only hold 200 individuals at a maximum. So this really emphasises the need to recover these species back to the wild as soon as possible, so that we can grow the population to more than 200 individuals and ensure that it's viable into the future. So we need to release them back to the wild, but where can we release them to? So this map shows different islands across the Pacific, and circled is Guam. So this is where the sea heck are native to. And this is our ultimate goal, to reintroduce sea heck back to Guam. But currently, the reason that they went extinct in the first place, the brown tree snakes, they're still present on Guam. So we can't reintroduce them back to Guam right now, because they will probably just get predated again. But we do need to recover them back to the wild as soon as possible. They've been in captivity for more than 30 years now. So we're looking at other sites that are free of brown tree snakes in other islands in the Pacific, including the Northern Mariana Islands, the Federated States of Micronesia, and then also Palmyra Atoll. So that's a US, it's a nature preserve that's co-owned by the US Fish and Wildlife Service and Nature Conservancy. So all of these island sites, they're no longer reintroductions. This would be an assisted colonization. So that's releasing the sea heck to sites outside of their native range. And these assisted colonizations come with potential risks of ecosystem impacts. So we're pretty concerned about the potential for these ecosystem impacts to happen. So to try and assess this risk, we've gone to the invasive species literature. And for invasive species, the way they look at the impacts of these species is to look at the different mechanisms of impact. And three mechanisms of impact in particular are of concern for the sea heck, and that's through disease, through predation, and then also through competition. So if we first look at the potential impact through disease, so to translocate a sea heck and release them to the wild, we would be taking individuals from a captive population on the US mainland and then releasing them to these island sites in the Pacific. So that would be crossing different geographical as well as ecological barriers. And that has the risk of bringing diseases which are in the captive population and releasing them to into those ecosystems where they might not be present. So to assess this risk, we're carrying out a disease risk analysis. So this is actually recommended in the IUCN guidelines for reintroductions and other conservation translocations. And this is currently ongoing work. And this is being carried out by Dr. Claudia Carrero, who's also at ZSL. And this disease risk analysis aims to identify all the disease hazards in the captive population, and also to look at the different species that are present in these in the ecosystems at the potential release sites. And for example, a disease hazard that it's identified is West Nile virus. So this is present in bird species in the US mainland, but hasn't been observed in birds on these islands in the Pacific. So that's quite a worry. And this disease risk analysis not only identifies these disease hazards, but also suggests mitigation strategies. So for West Nile virus, this would be putting in place robust quarantine procedures before any translocations could take place. So to look at the other mechanisms of impact that we're concerned about, predation. So this is because the sea hek are a predatory species. Their diet in the wild mainly consists of lizards as well as invertebrate species. So there could be predation pressure on these species in the release site ecosystems. And then also through competition. So there may be bird species or other similar species that use the same resources that sea hek use. So if we release sea hek to these new ecosystems, then they would be competing with those native bird species. So to assess this, we're using ecosystem modeling. So you can see here an interaction network. And this shows different species or groups of species in a potential release site and how they interact with each other, so shown by those arrows. And these interactions can be positive or negative or no interaction. And those interactions represent predator-prey relationships or competitive relationships, or also could be mutually beneficial. And you can then use mathematical modeling to model this ecosystem and to see the different abundances of these species with and without sea hek in the system, or whether those abundances, whether they increase or decrease with and without the sea hek there. So what's next for recovery planning for the sea hek? So our work so far has really emphasised the need to increase the total sea hek population size, and this is through both increasing the population size in captivity to that maximum of 200 individuals that they can hold, but also by recovering the species to the wild so that we can start growing populations in the wild as well. We also need to continue to work with all our stakeholder groups to decide our final release strategy. And we're hoping for our first wild releases in 2022, so after more than 30 years in captivity. And I'd just like to say thanks to the rest of the sea hek recovery team, as well as the wider recovery group, and also thanks to you all for listening.
Thank you very much, Amanda. That was a really interesting sort of insight into a specific programme and a species that you're trying to bring back into the wild. There is a question about releasing those birds back onto one of these islands, and whether you can maybe control some of the risks by releasing them into a fenced area or aviary at first to sort of monitor them. Is this an approach that is likely to be taken?
Yeah, so our first releases are going to kind of be like trial releases, and that's as much for the sea hek as for the ecosystem as well, because sea hek, they haven't been in the wild for 30 years, so we don't know how well they're going to do as well. So we're looking at really small island sites so that they're away from other islands to reduce the risk of them dispersing to other places. And these small sites can also mean that we can monitor the sea hek really closely to see how they're doing, but also monitor the ecosystem as well and monitor the species in the ecosystem. And that ecosystem modelling, one of the things that it can hopefully tell us is what indicator species we can watch. So what ones maybe would decrease in abundance first if that was going to happen. So we can pay particular attention and monitor those closely, so that if there is an effect that we're not happy with or stakeholders aren't happy with, we can put it we can have an exit strategy and maybe remove those individuals back to captivity or to somewhere else or something like that if there was really an unacceptable impact.
Oh, brilliant! Sounds like a very sensible approach. And in relation to the island where they used to exist, Guam, the snakes that you mentioned, the brown tree snakes, is that something that they could ever eliminate so that the sea hek could go back to Guam in the future?
Yeah, so there are like whole teams of people who are really working on this on Guam. So at the moment, they're looking at things like dropping bait with like a poison on it. So they've done some trials of that, and I think they have found some successes, but I don't think it's been that widely like used across Guam yet. They're also trying snake-proof fenced areas to try and like exclude the snakes from them, and then control snakes within those fenced areas. With Guam, there are like tropical storms and things like that. So there's potential like trees and things like that to fall on the fences all the time. So you really would need to constantly maintain the fences if you wanted to do that. And also for birds, there would always be the risk of them like flying out. So you'd have to be a very large like fenced area. Yeah, we are hoping that to reintroduce them to Guam eventually. So like maybe a combination of all these strategies, yeah, to try and, if not eradicate, then suppress the snakes to a level where maybe birds can still survive with those present.
Brilliant. Thank you so much, Amanda. We will bring you back later on for more questions. But for now, we're going to move on to our final speaker this evening, who is also from the Zoological Society of London, and that is Paul Pearce-Kelly. Paul is ZSL's Senior Curator of Invertebrates and Fish, and specialises in the development and management of species conservation, breeding and reintroduction programmes, with particular focus on ectotherms, also known as cold-blooded animals. Paul coordinates the European and global partulid snail conservation, breeding and reintroduction programme, and has been involved with their conservation for the last 30 years. So he's going to be talking now about reintroducing partulid snails in the wild. Paul.
Pleasure to join you today. I'm going to bring my presentation up, hopefully. Here we go. Okay, hopefully everyone can see that. All right, so yes, I'm going to be talking about, and by the way, I'm representing the work of many colleagues and agencies, and I'll give that as profile at the end of the presentation. But I'm talking about the fairly famous Partula tree snails from French Polynesia. A very well-known group of snails in terms of insights they're giving us into evolutionary genetics and how species evolve. And am I in my ability to go to the next slide here? There we go. Yeah. I hope everyone can see. Very well-known group. First, for a long time known to the Polynesians, who's part of our rich cultural heritage. To Western science, they were first made known on Captain Cook's voyage and have been interesting from a scientific perspective for a long time. A lot of work's been done. Unfortunately, they also gained fame as being a classic example of species really threatened by introduced predators, and just showing how vulnerable species can be when they're, for instance, on these kind of island environments where they don't have a lot of predators and native threats. And when these invasives can be introduced, they can really cause a tremendous problem. And that's what PASH have really shown. And there's one of them down here, the Euglandina dirosii wolf snail. So unfortunately—
Paul, I'm sorry to interrupt. We just had a slightly strange background noise there. So I think it might have gone now, which makes me wonder if it was me. But if you could just speak up, because it's fainter than you, so people should still be able to hear you. But yeah, if you just speak up, that should help. Thank you.
Yeah. Sorry about that, everybody. So just to briefly summarize, the African giant snail was introduced to the region, Tahiti, Moorea, French Polynesia in particular, became a major problem, agricultural pests. Then a predatory snail was introduced as a potential control, unfortunately without any real trials. And this predatory snail, the rosy wolf snail, became quickly found that it didn't like the African snails, the introduced snails, but it loved the actual native snails, unfortunately, and it set about wiping them out across a whole island group. So it caused a tremendous problem. A very large amount of species were lost. A little later, another predator turned up on the scene, the actual flatworm, the Platydemus flatworm, it's really one of the big problem species moving around the world. Again, sometimes due to biocontrol, sometimes just getting into regions by accident and causing a lot of problems to native snails and other species as well. So the current situation is a lot of reduced populations, lost species from those island groups, but fortunately some surviving species still survive. So you can see a map of what's left in the region, separate to our reintroduction species. And then in the conservation breeding program and the international and the stud books are managed by my colleagues at CEVERSEL and London Zoo and at the St. Louis Zoo in North America. And it currently records we're caring for 15 species and subspecies, the majority of which are indeed extinct in the wild. And they've been in the conservation breeding program for the best part of 30 years after a classic species rescue effort to try and keep ahead of what the predators were doing in terms of wiping out species. So it's a classic ARC type situation for trying to respond to a species crisis. And for the last 30 years or so, they've been in the breeding program. We've had to understand the husbandry and care requirements and develop the dietary needs and develop best husbandry practice. And if you're going to keep a partula snail alive, you need to keep the enteric fauna fauna flagellate populations going inside so they can still digest cellulose, etc. So you're trying to conserve not just the actual species you can see in its tank, but also what keeps that species going as well. I should also quickly just make a quick point that we've learned a lot from working with these snails, and it makes the point that things aren't in pigeonholes. What you work with one species can benefit many other species as well, and partially a good example there. And they've really shown us how you can manage species which have to be kept as populations when you can't track the individual. So they've really been a really great aid for doing that, and then we can apply that knowledge and those management techniques to a whole range of different species when you can't track at the individual level, working with colleagues in the conservation breeding specialist group, etc. And with the reintroduction phase, we actually started thinking about island forest reserves, protected areas, really, as you can see on the left here, little plots which are predator proof, keep the invasive snail out, etc. And we did the first trial back in 1994. Things moved very slowly in the snail conservation world, and we did show that it could be done, but it was quite intensive effort. And fortunately, because of the ongoing field work that was being done and the observing how the predator dynamics were going, we could see that we could move to working with just natural forest areas where the populations of the predator threat had reduced or completely gone. So we had that evolutionary approach, intensive releases in very close reserves to this more open system. And just as we heard earlier, if you're going to reintroduce anything, you don't want to create more problems. So putting some of these species back, we had to go through the disease risk analysis. Are they still healthy animals? They're not going to create problems for bringing pathogens into the region, etc. So again, veterinary health screening and research is extremely important in these areas. And we heard about releasing species outside of its natural range. Well, all of our program partula snails were looking at trying to release them in their known ranges. But again, because of some challenges with particularly the flatworms, questioned at least in the early stages whether we could do that into known areas. We also did a study, again, following the IUCN best practice, for if we couldn't put a species back because the predator threat was so great, are there areas where species now lost had ecological environments which the target species could actually thrive in? And we went through that process working with Dr. Justin Gerlach, and we found that yes, we could find alternative options if we had to. So we went through all of those planning stages. Then the reintroductions themselves started, and started in 2015. And we were very, and you're moving animals from one part of the world to the other, so it's quite a challenge in its own right. And zoos all collaborated in providing the snails to be able to do that. We had quite high mortalities on that very first tentative release trial we did. And over successive years, we've really reduced the actual mortalities now to pretty much zero. So a very long international transport system, we've really shown that we can get very high results and have very healthy animals coming out of the other end to go through the quarantine period with the French Polynesian government's environmental department before they go to the forest. And then we heard earlier about selection and are we still producing fit for release animals, you know, are they viable? So here are our snails marked up, ready to go into the field, different species. But of course, over 30 years, have they just grown used to being in a zoo environment, in lab type conditions, etc. So the proof is in the pudding. So how do they behave when they go back into the field? And it's been quite heartening to see that they are really quite robust little things, and it's been heartening to see that the dispersal rates seem to be very healthy. And should point out that when you're talking about many different species in a group like we have with partially, you get some species doing incredibly well, some so-so, and others we're much more worried about. So you need to have the fuller picture. But as a general summary, we've been very pleased to see how these snails respond back into the native environment. And you can see here's a range of species just so you can show over the various years of the releases, how they can revert back to natural balance, etc. And again, the follow-up work, all of the collaborating zoos working with the French Polynesian government have for years contributed to a field biologist being on the ground the whole time, working with the environmental department and local communities, etc. So that's enabled us to understand the predator dynamics, the threat realities, and how they've abated on down now. As a classic picture, you see a predator will go into a region. If it has no competitors and no natural controls, it will expand and become a really tremendous problem. And that's exactly what we found with these predators. And then over time, the predator threat reduces, other pressures come in, and you get to this more natural balance. And now with the main predator focus, we really struggle to find its presence on most of the type of animals, which is very heartening. But we only know that because we've been doing the monitoring.
Paul, you're dropping off ever so slightly, and sometimes towards the end of sentences. So yeah.
So just to summarise here,
Thank you.
14 species and subspecies, over 15,000 individual animals have been released over the last 5 years onto several of the key islands, Moorea, Tahiti, Huahine, and we can track the mortality by looking for shells, dead shells on the ground, so it gives us a rough idea, again with very close scrutiny in the field, gives us an idea of fairly optimistic survival. Again, it's the best we can do with the environment we're monitoring in. And that gives us an understanding of some species doing very well. Some, well, yes, not quite so well, but we can see there's every reason to be kind of hopeful. And then some more concerning, perhaps because they're slower to disperse. One species in particular tends to go down onto the ground, whereas the rest of these tree snails to go up, which is exactly what we want to see if there are any predators within an environment. Whereas one of these species likes to go down, so again, it's a very worrying outlook for that particular one. So we may have to be caring for that one in the long term. Challenges, well, very complex, high forest canopies. How on earth can you see what's going on when you've released your snails and they've all gone up? So that's an ongoing challenge to better understand what's happening with the field side of things when you're trying to monitor a population. Helping additional species, absolutely a key concern we have in terms of there are always more species requiring help, and this is one of the famous species from Guam, which is Crocodile gibber. And a couple of years ago, the French Polynesian government, the Department of Environment, held a very important workshop involving representatives from all of the Pacific region. And we learned a tremendous amount in terms of the realities on the ground, not just for any particular target species, but for all of the mollusks which are known on those terrestrial systems. And we had an actual important statement from that workshop, which identified in the region of 6,000 terrestrial species of land snails on over the Pacific region. So we've been working with a tiny subset, but if you look at the bigger picture within that region, you can see just how many species are very likely requiring conservation help, very similar to what we've just seen in the case of partial care. Colleagues have alluded to climate change, some of the other challenges. It's said that there are no climate skeptics in French Polynesia because they see the effects on the ground, the changing seasonalities, more extreme weather events, etc. That's something we have to factor in terms of viable places. And that highlights the need for biobanking. Every species, I hope, including the northern rhino, let's hope, is actually represented at some degree in cryopreserved states. So there's no reason now why that can't be done for the majority of species, but it probably has vast gaps in terms of how that's currently done. And we've lost partial species over the years, but at least some of them, which we have lost, reside in these biobanks, so all is not completely lost. But summarising the key success factors very briefly, partialists had a successful, hopefully successful outcome so far, because they were well known. People knew about them. There were lots of research going on, collaborations. There was an early alert of the unfolding threats they were facing. There was a receptive conservation response. I must highlight the work of the Conservation Planning Specialist Group, the International Zoo Community and the French Polynesian governments. They were all committed to trying to do something about it. There were key engaged people. There was a really robust but flexible action plan produced and best practice protocols. They needed relatively modest funding, which is a big help, but more importantly, that funding was sustained and it was kept going by international collaboration, again with the French Polynesian government and the international zoo community, and it needed a good dollop of luck as well. And just to end by highlighting and giving credit to the wide range of people involved in this effort across French Polynesia, the government, the local communities, the biologists, etc., the ex situ side, the collaborating zoos, which kept those populations going and provided the resources for the on-the-ground research and collaboration with biologists. A wider range of specialists across the international community sharing knowledge and really making a highest chance of outcome by this collaboration. No conservation initiative really can survive, can really be viable unless we have this degree of collaboration. And most of all, just to end, to give credit to our wonderful colleague, Dr. Jobber-Keith, who very unfortunately just recently died from COVID-19, just to show how serious that is, after 20 years working in the field as the field program biologist with the partial program and that collaborative consortium. And he had just retired, and part of his work was working with colleagues like Robert, who's taking on this work across the Pacific, across the partial region in French Polynesia. So very sad, but we dedicate this presentation to Trevor and look forward to the work that he's helped with such a wide range of colleagues to hopefully have a successful outcome. And that's our summary of the partial program.
Thank you so much, Paul. And apologies for anyone watching. We've absolutely no idea where that strange sound is coming from in the background. But that was a really interesting insight into that work, Paul, and sort of the years of effort that have gone into it as well. There is one comment which I'd like to share with you that's come through. It's from somebody who's watching in France, and now I'm not going to be able to find it because it's been upvoted somewhere, but basically someone was here we go. They're watching from France. There is no French zoo working for on the partialist snail conservation program that they're aware of, and it is not a topic that they know of in France, despite it happening in Polynesia. So they say thank you so much for the work. So that's really nice to sort of be raising awareness of these incredible animals and the work you're doing. What's the next step for the program, Paul? Because you've obviously had quite a lot of success, but where does it go next?
Yeah. Well, thank you for that. I should point out that Thoiry Zoo in France worked for many years as part of the program, and Zoo Bouval is just about to join the program. So France is doing its bit.
Excellent.
The reintroductions continue, and it's very hard to say, is any conservation endeavour successful? How do you ever gauge success? But one of the most obvious indications is the population still persisting after your efforts? So that's our ongoing challenge to prove not just short-term persistence, but long-term persistence. And I'd be amazed if it's going to be successful for every species we have in the program. But let's keep our fingers crossed that as many as possible will actually have a successful outcome. But we envisage at least probably we think we're maybe halfway through the reintroduction method. And then hopefully you can take a lighter touch and just keep that reintroduction element going when it's needed, but it's not such a big deal, and it's everyone's more in a kind of they're kind of used to working with the system, etc. So that is our hope. But you never know. And again, climate change and the rest of it could change things very much, and something else could turn up. So you need to be flexible and always looking out. But how you would apply this conservation plan across those hundreds of other species, those thousands of other species, extrapolate that to a global perspective and tens of thousands of species, and it's a challenge. So you take what you can from the practical things that work, and you hopefully adapt them accordingly to a very changing world, which we find ourselves.
Absolutely. Well, let's bring back the rest of this evening's speakers because we have still got quite a few questions. So we're going to see how many more we can get through in the next 10 minutes. The most popular question at the moment is about the risk of reintroducing an extinct species in the wild, and the sort of the risk this poses to that ecosystem and other wild species. And Amanda, I know that was something you touched on with the CHEC, and obviously talking about how important it is that you're aware of this and you do everything you can to avoid that happening. But I mean, I imagine that's the case then for all kind of reintroduction or introduction programs, that it's something you're always having to think about. I'm not sure, Amanda, if you know of any cases perhaps where that those risks sort of have become apparent.
Well, so you know, there are definitely cases where things have gone wrong when they've put a species not as a conservation translocation, but as generally a translocation. So for example, like cane toads in Australia. So yeah, so there's, you know, invasive species like where they where species went into a novel ecosystem and then have done, you know, had really negative impacts. But from a conservation point of view, there's probably other people, like maybe some of the other panelists know, but yeah, I don't know any examples where they did a conservation translocation and it's gone wrong. So, and yeah, I think definitely with the IUCN guidelines, they're really advocating for doing these risk assessments really carefully, which is, yeah, and what we're trying to do, and expanded even beyond those guidelines. So, you know, with ecosystem modeling, there are all quite new techniques that we're using to really understand the risks.
Yeah, really important. Axel, another popular question that's come through, which you may or may not be able to comment on, refers to, in South Africa, there are a number of the number of white rhinos has increased since farmers started breeding them. Would it make sense to try and reach out to the farming community for other species as well?
Basically, conservation is something that should not only be in the hands of scientists, if you will. We should look for, you know, collaborations across sectors, whether that, you know, involves other kinds of landowners or stakeholders. So we should never close the door, and indeed we should open more doors. I don't know about those efforts specifically. We need to also make sure that everything that is done is you know, at the highest quality. But one example I think that is interesting is that across Southern Africa, for instance, you know, a lot of ranching type communities turn towards more wildlife reserves in a way that has provided connectivity even among the different national parks. And so there's definitely a way to find win-win solutions, I think, for species and for local people too. And certainly I think this is the case, and maybe back to the questions of people, you know, planting plants in their gardens and such. There is an opportunity, I think, if we just explore and if we just use our imagination to get a greater buy-in across the different kinds of communities in the world.
Axel, very quickly, there's another one that came in specifically for you from a teacher saying they're looking for a comprehensive list of species that have been saved due to human intervention, such as captive breeding programs. Is this a list that would be available through the IUCN? Are you aware of any such list that this teacher could find?
Yeah, very good question. And it's interesting because in the early days of the IUCN Conservation Translocation Specialist Group, we tried to stay on top of basically all such translocations. But then it became unwieldy because there's so much actually going on. I would just really go to the IUCN case studies and IUCN-CTSG.org, because you'll see not only the kinds of species that are being saved, but a success evaluation among them. Okay, and so basically you can see how well people have done in achieving the objectives that they set out themselves. So I think that's one resource, and in the same place as well, there's a searchable database for those specific species that'll get you at the least a good head start with at least 400 species to get you sort of started with the students. And by the way, thank you for doing that. We need you, and we need those future generations to help carry that torch.
Brilliant. Thank you, Axel. Sarah, and another question that came in for you is, with climate constantly changing, does it cause a concern that if you reintroduce a species to its indigenous range within 10 or so years, it may no longer be a suitable area anymore?
Definitely a concern, yeah. And again, it's something that Joe, who I mentioned, who's my PhD student, has been working on, both in terms of looking at whether climate suitability explains the failure of translocations, and we can find a climate signal. So where translocations have failed, they are more likely to be in less suitable climate. But also he's projecting into the future and showing that these are only predictions, it's like a weather forecast, but over longer time scales, where we take those projections and we look into the future, then the sort of take-home message from his work is that if we're reintroducing things that are at the sort of warmer end of a species range, those translocations are less likely to survive into the future. So when we look at the projections, we can say, well, actually, the ones that are sort of, in the northern hemisphere, it would be further north, and the southern hemisphere would be further south, or it might be elevationally higher. We could sort of summarize that they've got better prospects than the warmer sites. So yeah, it's definitely something that I think the conservation research community is definitely getting on board, because we use all these very competing intensive models, but it's not necessarily translating into practice yet. And that's what me and Joe have been trying to work on recently. So
thank you. A couple more I think we can squeeze in just before the end. Paul and Axel, you might want to comment on that one. On this one, how do you bring institutions with you for recovery programs that require long-term commitments? It can be very hard, as most extinct in the wild species are not box office species. Paul, I imagine perhaps this is maybe a question people might have raised in the past about parchula snails. Has it been an issue?
Well, I'm pleased to report that my experience is, providing you can just communicate accordingly. Things like snails and crickets and beetles and things, they're quite capable of capturing our imagination and our support across a wide range, local community, collaboration organisations to work with them, etc. So they're all fascinating. They all have that ability. It's just a case of getting that communication out and indeed collaborating with them. I think the key success, no matter what you're looking at, is there's some degree of awareness and there's some real collaboration going on. The challenge is what you can do in terms of when you're faced now with the numbers of species requiring help. So if you look at, say, the world's corals, which we're looking at the moment, you're really looking at a situation where in very short order, all those coral species need to come into some kind of culture care, such as the degree of threat we're looking at in the field. But how you would be able to see reintroductions when you're changing the chemistry of the ocean, thermal stress, and half a dozen other stresses. This shows, the scale of the actual. So facing that, everyone is a conservationist, because to save those kind of species is absolutely all of us. You could do professional front-end work with the species, but society as a whole has to be absolutely behind wanting to have an environment where those species can have some hope of long-term persistence.
Well, let's finish on a positive note. Amanda, one question which came through referred to the oryx that actually Axel spoke about in the first presentation, and it was the question: at what point is a species no longer extinct in the wild? Is it when you first introduce or sort of relocate a species outside of a captive environment, or is there some other indication that that you no longer classify it as that?
So Axel might be able to be better at answering. So there are definite IUCN guidelines for when a species can be classed as not extinct in the wild anymore. So, for example, the Koko or Guam rail has recently been downlisted to critically endangered, and that's because so they need to be reintroduced back into the wild. But I think it also has to be within their native range, I think, for it to be classed. Because with the Koko, they had originally tried to rescue them to a different island, which actually has a larger population than the one that was used to qualify for them for downlisting. So I think it needs to be in the native range as well, but maybe I'm wrong. Maybe check with Axel.
Are you any clearer sort of on at what point you can say that?
Well, I love the question, because it's a question that we asked about a year ago, actually, in advance of submitting the IUCN motion. And one thing that is really great is that the IUCN red listing team, including some key staff from ZSL as well, have made newer changes where you use basically probabilistic modeling, first of all, to see if something might be extinct or extinct in the wild, but which in some ways can be used in reverse as well, to go from extinct in the wild to critically endangered. It is a step that isn't as well described as steps between the other threat levels, but I think it's on the right road, using the right demographics and sort of in alignment with, I suppose, what our task force is trying to do. So it's a great question.
Fantastic. Well, thank you all so much for your talks this evening. It's been a really interesting insight into the subject, and thank you also to all of you who have been watching and sending in your questions as well. We've really appreciated receiving them all. I'm going to share my screen with you one last time, just so that you can see that SurveyMonkey link that I mentioned at the beginning. Please do let us know what you thought of tonight's event. We'd really like to know. Just go to www.surveymonkey.com/r/ZSL event 6. That's surveymonkey.com/r/ZSL event 6, and you'll be able to share your thoughts with us there. We do hope you enjoyed this evening's event. It is one of many that we are running at ZSL. So if you'd like to find out about upcoming events, then do go to our website, which is zsl.org/science/events, where you'll be able to find out about our next evening event, which will be in May. We're taking an Easter break over April, but we'll be back on the 11th of May with Why Do Eggs Fail? And in the meantime, if you're still looking for a science fix between now and then do tune in for our Wild Lunch Wednesdays as well, which are taking place fortnightly. And to make sure you don't miss any of those, press the subscribe button on our YouTube channel. As always, you can find out more information about our work on our website, and also ways that you can get involved and help support. So please do have a look there. But for now, thank you for joining us. Bye-bye.